Hello, I'm Tom Martin from the University of California, San Francisco, Helen Diller Family Comprehensive Cancer Center. And I'm joined today with Blood Cancers Today Editor-in-Chief, Dr. Sagar Lonial of the Winship Cancer Institute in Emory, Dr. Shambavi Richard of the Center of Excellence for Multiple Myeloma in the Icahn School of Medicine at Mount Sinai in New York, and Dr. Peter Voorhees of the Levine Cancer Institute in North Carolina. And this is our first roundtable of discussion on BCMA-targeted CAR-T cell therapy. So now we have some therapeutics that are actually approved for use. And in fact, the two therapeutics that were first to be approved were both CAR-T cell therapeutics, both actually targeting BCMA. The first one came from the original studies using the CAR-T cell that was noted as BB2121, and that eventually became our product known as Idecabtagene Vicleucel. And Dr. Voorhees, can you give us a little update on what's happening with Idecel and how we got to the point we are right now?
Yeah, no, absolutely. So as Tom mentioned, Idecabtagene Vicleucel is a BCMA-targeted CAR-T cell therapy, and the phase 1 study was published in the New England Journal of Medicine back in 2019 by Nupur Rajay and colleagues. And what they did in this portion of the KarMMa studies is they looked at anywhere from 50 to 800 times 10 to the 6th CAR-T cells per patient. Patients received lymphodepletion chemotherapy with fludarabine and cyclophosphamide from days -5 through -3, and then they underwent a CAR-T cell infusion on day 0. And what they found in this particular portion of the study is that dose mattered. So overall response rate was only 33% for those that got 50 times 10 to the 6th CAR-T cells, whereas those that received 450 to 800 times 10 to the 6th CAR-T cells had response rates of over 90%. And in this relatively small number of patients, median progression-free survival was close to 12 months at 11.8. Small numbers of patients, but there was a clear correlation with the number of cells that you got with regards to response, CAR-T cell expansion, whether that was measured by Cmax or area under the curve, whether you got cytokine release syndrome or not. And there was also a signal towards worse overall responses for those that had high-risk cytogenetics versus those who had standard-risk cytogenetics. So that led to the phase 2 portion of the KarMMa trial, which was also published in the New England Journal of Medicine 2 years later in 2021. Nikhil Munshi and colleagues published this data. And they took patients who had relapsed refractory multiple myeloma. They had to have had at least 3 prior regimens, including an IMiD, a proteasome inhibitor, and an anti-CD38 antibody. They had to be refractory to their last line of therapy. They had to have a good performance status and a creatinine clearance of 45 mils per minute or better. And patients received anywhere from 150 to 450 times 10 to the 6th CAR-T cells in the study, the vast majority of these patients receiving either 300 or 450 times 10 to the 6th, and the lymphodepletion and the timing of CAR-T cell infusion was the same as in the phase 1 trial. And what they found in this particular study is that the overall response rate was 73%, with a third of patients achieving at least a complete response. The median progression-free survival for the entire group was 8.8 months, but when you single out those that got 450 times 10 to the 6 cells, median progression-free survival was 12.1 months, and median duration of response was 10.7 months. So this, I think is really an impressive data set, and that's what led to regulatory approval of this particular regimen for patients with relapsed refractory multiple myeloma. They also looked at markers, measures of response, and what they found here is that, for those patients who had a low dose of cells, response tended to be negatively impacted by that, and patients who had revised ISS stage 3 disease going into CAR-T cell therapy also had inferior response rates, but they saw very impressive responses in triple and penta-refractory disease, patients who had extramedullary disease as well. So really nice data.
Awesome. Do you think that in terms of the expression on BCMA on the surface of the plasma cells, do you think that that is important, or is there any data that suggests we should start looking at that clinically?
Yeah, so far, no. So in the initial phase one portion of the KarMMa trials, what they found was is that whether you had more than 50% BCMA expression versus less, that your responses were very good. Now they're dichotomizing a continuous variable. So, someone who has 5% expression versus 95% expression, maybe there's a difference there. I think we need to study that more carefully, but, the bottom line is that you can respond with a wide range of BCMA expression levels.
Excellent. So Idecel, is certainly it was the first drug to be approved in this setting and has certainly been used clinically. And we do have some real world experience that's been, Doris Hansen has presented at multiple conferences, the real world experience, and it looks like it performs just as well as what you said, right? And I think clinically, we're all seeing the same thing at our own centers, that it does work. It works in some of these patients who you wouldn't expect to have really any significant response for any other drug that we would have had access to, right?
Yeah, it's pretty rare that real world experience recapitulates what you see in phase one and phase two testing. So the fact that the real world experience, was virtually identical with regards to progression-free survival is actually very reassuring. And what we found in that particular real world analysis is that prior exposure to BCMA targeted therapies did negatively impact progression-free survival. And there was also a signal towards inferior progression-free survival for those patients who had high-risk cytogenetic features.